Hi, I'm Steve Cassidy from the Keiser lab at the University of Florida. Thanks for coming to my talk. Today I'll be talking to you about social parasite exposure and disease defenses in acorn ants. Eusocial insect colonies, such as ants, have disease defenses at the individual and group levels. This figure here demonstrates this by showing how workers in ant colonies, shown on the right in blue, protect the colony, or the superorganism, using behaviors in a similar way that immune cells, shown on the left in blue, use physiology to protect individual animals. Individuals within the colony, of course, also have their own innate physiological immune systems, but they use social behaviors as a second layer of protection. This is known as social immunity, and there are several layers of social immune protection that include collective hygiene, sanitation, infection removal, and social network modulation. At this point in the pandemic, this concept is probably pretty straightforward for everyone, as we've all been performing these behaviors for over a year, such as mask wearing and social distancing. For the ants in our study, we'll be focusing on collective hygiene. We have previously shown in our acorn ant system that these two levels of immunity seem to be correlated, such that individuals with stronger social immunity consist of workers with weaker individual immune systems. This is shown in this figure here, whereas individual immune strength increases on the x-axis, social immune strength decreases on the y-axis. In other words, acorn ant colonies either have strong individual or collective protection, but not both. As a follow-up to that study, we wanted to investigate how an antagonistic interaction with another ant species may affect both levels of disease protection. The specific question we asked here was, to what degree is multi-level disease protection affected by exposure to an antagonistic stimulus? This will help elucidate if antagonism can alter disease defenses. To investigate this question, we used Temnothorax curvispinosus, a common ant species throughout North America that has relatively small colonies that live in hollowed-out acorns, like you can see in this picture here. To test their immunity, we exposed them to a generalist entomopathogen, Metarhizium robertsi, which is a fungus that inhabits soils around the world and has a huge host range and is a known parasite of our Temnothorax. The socially parasitic ant we used to expose our host colonies with was Temnothorax americanus, seen in the foreground of this picture here. This is a very well-known ant species that send americanus workers to invade host colonies, steal their brood, and return to their own nests, where the brood eventually mature and behave as if they are part of that parasitic ant colony. This is a very costly interaction for the host colonies, since most, if not all, of their offspring are lost. This brood theft strategy is relatively rare in ants, but species that do this have generally been thought to have closely evolved with their host colonies that they steal from, such as the curvispinosus that you see in the background of this picture. For the colony to find and raid host colonies, individual americanus scouts go out, locate the colonies, and return to the parasitic colony with the location information. And due to the coevolution, we expect that exposure to a single scout may influence the physiology and or behavior of host species exposed. These are commonly known as the slave-making ant, and I really hesitate to even use that term due to its obviously problematic nature, but this is how they are widely known. Even Darwin referred to them as such in The Origin of Species. And I think this is a harmful term and actually doesn't really describe what's happening very accurately, and therefore shouldn't really be used. I use the term social parasites since these ants use social behavior to parasitize host colonies in order to increase its own colony size. But there are several different alternative terms that you can use to replace that with, some of which I list here. To measure the impact social parasites have on disease protection, we measure the baseline individual and social immunity of curvispinosus colonies. Then we expose the entire curvispinosus colonies to a single americanus ant until the host colony successfully killed the scouting parasite. Finally, we measure the same individual and social immunity traits after social parasite exposure to see if either of these disease defenses changed. To measure individual immunity, or within colony immunity, we measured a proxy for immune strength of worker ants within each host colony by exposing them to fungal pathogens and measuring how long they lived once infected with the fungus compared to uninfected workers. We did this on 96 individuals before and after exposure to the scalp. To measure social immunity or collective immunity, we measured the latency of corpse removal, which is a measure of collective hygiene, in the respective host colonies. We did this across all 16 colonies. Finally, we exposed only half of our curvispinosus host colonies to the americanus parasites. We measured individual immunity by exposing 3 workers to fungal spores and 3 workers to a control solution from each colony. We calculated this relative value of immunity by dividing the time to death of infected individuals by the time to death of uninfected controls. We measured corpse removal by freezing uninfected workers to kill them, placing them one dead worker within the colony to see how long it took for that host colony to remove that corpse. And it's important to remember that faster time to death indicates weaker individual immunity, whereas faster corpse removal indicates stronger social immunity. We found that being exposed to a social parasite increased host workers' individual immune response. This is shown in this figure here, where smaller points represent the relative immunity of each host colony. The colors and shapes represent different antagonistic treatments. The larger points represent the average for those treatments, and the dotted line represent the point at which experimental colonies were exposed to the social parasite. The relative immunity of individuals in colonies appears to increase after exposure to these parasitic scouts, whereas individuals from control colonies had about the same immunity. This was a bit surprising to me, as stressful interactions such as antagonism can generally decrease immune function across other animal systems. However, for social immunity, we did not find evidence that the social parasite exposure affects how fast colonies remove corpses from their nest. You can see that in this figure here, which is pretty similar to the previous figure, the main difference being that we measured corpse removal twice before and twice after exposure. And regardless of treatment, corpse removal remained relatively consistent across all treatments. Our results suggest that exposure to a dangerous antagonist may affect each level of the multi-level immune system differently, and that some antagonism may in fact increase immunity of individuals, but does not lead to an increased collective immunity. And with that, I would like to say thank you to all my co-authors, especially the amazing undergrads and my advisor, Nick Keiser, as well as the University of Florida for supporting our research. Thanks.